We've all been inspired by mentors, and Judith Folkman has been a mentor to me and my brothers. And we will talk about that, but in preparing for this, we think about angiogenesis and the biology every day. So I just want to start out, I'm a clinician at University of Pennsylvania, and this is a new member of the club, a nursing student, is 23 years old, and she had an auditory hallucination. Okay. So I'm technologically challenged here. Lauren? Anyway, a nursing student, you could read. Maybe. There you go. There you go. Thank you. Thanks, Eric. You're much better. Okay, thank you. That's right. That's why. Eric, you need to come to the operating room. So you can see there's not much angiogenesis here, maybe a little patch, and this is probably, hopefully low grade, but it might be AA. And here, right in Dr. Del Mestro will recognize this as being near Wernicke's area, but she didn't have much speech problem. And then we do all this beautiful tractography, and we can actually get to the white matter in a way that probably Penfield couldn't, because he didn't have all this wonderful imaging. But we did all the cortical mapping, and she emerged with some word finding difficulty, but she'll probably be okay. And so that's the current state. Now, we know that this is a continuum. She's only 23. And so one thing we learned from Folkman is to ask questions. Folkman's genius had many aspects of genius. But one, he said, is that as a surgeon, we are privileged to see in vivo biology and start asking questions. I'll always ask questions. So what is the difference from this and a glioblastoma? One is angiogenesis, two, invasion. I'm sure Raleigh could give us 10 differences, but there's a progression here. And the key thing with angiogenesis is, in Folkman's genius, was he was talking about molecular switches and pathways when no one used that vernacular. It's like he discovered a whole language before people even knew it existed. And so he could look at something like this and in his mind think, this is not just a tumor, an object that I'm going to remove safely, but think about its underlying biology. Now, this was also Monday, my second operation, and thinking about this talk, this you will recognize is glioblastoma. And this is from an Army colonel who had just marched in with his family at the Race for Hope in Philadelphia on Sunday. And he had 4 years ago a craniotomy in South Carolina, and then later had a biopsy in the frontal lobe. And both those procedures confirmed anaplastic. So we're going from a 2 to 3 that has now become a 4. And you can see all the hallmarks here, and I think you know this too well. Increased angiogenesis, blood flow. So the secret is the red button, yeah. Okay, well you can see the increased blood flow in this. You can see the pointer. Raleigh, you're a real friend, forever friend for life. Got it. Green light. Thank you. Thank you. Thank you. Thank you. Thank you. Okay. So here's necrosis, angiogenesis, invasion. You look at the Raynaud view, the T2 FLAIR, you can see this on the brainstem, so we decided to take out the anterior temporal lobe. There's so much discussion about gross total resection, and obviously this is not one we're not going to get a GTR on, right? Maybe in Montreal, probably, but diencephalon going down to the brainstem and so on. So this is one we really do need a biological answer for. And again, we're looking at the corticospinal tracts now and can sort of see the limits to our resection and so on. So why even operate on a patient like this? The patient was seen at Hopkins, and one of the neuro-oncologists said don't operate, but it had grown a lot even since the Hopkins visit, and he's beginning to develop cognitive changes. And in our brain tumor conference, we said, you know, we're in the era of precision, personalized medicine, and we need to get the markers. And Raleigh, you're going to talk about the methylated promoter. We all know about IDH-1, EGFR. And we have a very exciting immunotherapy trial that has opened IRB, and at Penn, we'll also be opening at UCSF with Sue Chang. It's Carl June's work on the chimeric antigen receptor, and Don O'Rourke is our principal investigator, surgeon, scientist. So we need to phenotype this tumor. We would need to do as much as we can because there is a possibility of proton therapy or other radiation, hypofractionation, and the radiation oncologists are telling us we should debulk the tumor because we don't really want to give whole brain therapy. Also, Dr. Desai and our team is the principal investigator for the NCI-sponsored ABTC, and there's a 1202 trial on one of the checkpoint inhibitors. And then we're also going to hear talks about electrical fields. So we need to know the diagnosis. This is not an AA. And so there's all these therapies now. And this came out last week, Carl June and Dave Porter's group. Karen, like you said, we're a little bit out of the box here talking about leukemia. It's a little bit outside my main expertise. But the liquid cancers have shown the way for solid tumors. Solid tumors are a little bit more complex for reasons we all know, but the liquid tumors, we now have incurable leukemia. This is from that paper. 2 years out, over half, these are censored data, there's a tremendous survival curve, which was not predictable even a few years ago due to these advances in immunotherapy. So we're very, very excited by all that. And so we've heard about cures and turning points, you know, and I think this could be it, because sooner or later, this is the reverse of the boy who cried wolf. One day this is going to be it, that we will find you know, we've felt like we're on the verge for so long. And I think one of the things that plagued Judah Folkman at the turn of the century is with Jim Watson, who was no shabby scientist, discovered DNA himself, and with Francis Crick, predicted that there would be no cancer in the year 2002. Well, that was a little, even for Judah Folkman's enthusiasm. What Judah Folkman actually predicted was that cancer would be a manageable disease, the way today HIV is a manageable disease, with combination therapy. That's what he predicted, and he wrote about that in Nature and so on. He never thought we would totally cure it. And so these are, this is young Steve Brem, very happy to be at Harvard with Dr. Folkman. And one of the things that Dr. Folkman, even though he was often the brightest person in the room, he didn't necessarily have to, and he had sort of your humility, Karen, and he would always get the best minds. And he told me, we need to know more about angiogenesis, Steve. And I didn't know about Raleigh Del Maestro was studying endothelial biology in England at the time, but Ramsey Cotran was the pathologist. He was the guru at the time. And so Ramsey Cotran later wrote the textbook on pathology. And this is just how our family has grown, the field has grown, but this is me back in the, what'd you say, 60s, 70s, the Beatles? Yeah, I had much more hair, which is, Judah Folk never had a lot of hair, but he always told, you know, it's what's beneath the scalp. And Henry has a lot of hair, but he's also in this Mick Jagger look, this kind of Rolling Stone look. And Henry today is the Cushing professor, and they've actually, the Brem family's been invited because they're creating a Henry Brem professorship at Hopkins. So Henry has certainly developed polymers and angiogenesis and experimental therapeutics that began at this era. And my youngest brother, Harold, actually grew up in this era. Here he's about 8, here he's 20 years later. And so for about 20 years, we always had a Brem in Folkman's lab. And Judah Folkman was always envious of that. He would write recommendations saying that not only is he hardworking, talented, and so on, but he has a network of brothers. This was before the era of Dropbox and Facebook and so on, so that we would be sharing ideas that would be, sort of. Judah Folkman was very generous. He gave me the credit for the first translational work in angiogenesis, basically to study and quantify angiogenesis. And we predicted that brain tumors would be the first to be amenable to anti-angiogenic therapy based on their biological vascularity. As you know, in our chapter, there's so many other historical and economic and demographic reasons why that didn't occur. But brain tumors took about 5 years more than colon cancer to get FDA approval. And Al Masella could also speak about all the barriers. And there's a book called Truth in Small Doses about how single labs and single companies produce single products. And so here we are. And this is years later when I showed Judah Folkman the first chapter, the first time, and his picture, which was sort of a historical moment because he was giving the couch lecture. And the 5 previous lectures had all received the Nobel Prize or were Nobel Prize winner. And at that time, Judah Folkman was very much in the mix to be the Nobel Prize winner, and he could have, should have, would have received the Nobel Prize, but he didn't. And that was the one word that sort of switched him from being very happy to not such a happy person. But I had a couple of days with Dr. Folkman back then, and that was the year 2006. And so I asked him sort of what was the secret to his genius, because who knew one day I might be giving a talk in front of a group of people. And he said it came from his family. His father was a rabbi, and they would have this thing every day at dinner where he'd always be asking questions. What is the meaning of this? How do you know this is really true? How would you test this? What do you, so he thought that this constant asking questions is sort of out of the box that he had as a child. He also had a passion for science, obviously, and he had some great mentors himself. But this mindset of always asking questions. So this was a cartoon that is New England Journal of Medicine, a classic paper in 1972. I came to the lab, and Judah Folkman was very generous. He let me write this chapter in clinical neurosurgery, single author, but it was really the concept of a switch, that tumors would go from avascular to vascular. And then later it was in Montreal where we actually found the first, probably a poor man's Avastin copper depletion, and that we actually demonstrated the truth of this. And here's this very serpiginous, hypervascular rabbit with a carcinoma. And then after we gave anti-angiogenic, we normalized the vessels. This is now Rakesh Jain, very elegant theory about normalizing the vessels. And we also observed that we proved the Folkman theory that tumors would remain as tiny nodules without blood vessels, but we also showed vascular cooption. We observed that, and that has been one of the reasons of one of the mechanisms of escape, as you know, and infiltration and invasion. At that time, Raleigh and I were working on anti-angiogenesis in Canada. We actually thought that if you could block invasion and angiogenesis, you would block both because they have very similar biological similarities, but they're actually different hallmarks of cancer, and it turns out that we've targeted VEGF, and as you know, VEGF is pro-angiogenic, but also maybe a negative regulator of invasion. And at this meeting, there's going to be a new, from Rolf Scherberg, am I pronouncing his name correctly? Rolf Bergvik is going to present another switch that when you block angiogenesis, you create a metabolic anaerobic switch. And he's got some beautiful paper on that and powerful data, and that's a good talk to go to. So the field really grew. Folkman used to joke back in the 70s and 80s, if you worked in one or two labs, you probably knew the whole field. And now it's, and we stopped counting here about 10 years ago. But even at this meeting, as Dave Reardon has said, angiogenesis research has dominated the landscape of neuro-oncology. So it's affected many other fields, and it's actually totally transformed ophthalmology, for example, vascular biology, obviously, and other diseases. So Folkman is true genius. Again, laboratory models predict clinical outcomes. Way back in 2000, this paper by Rubenstein showed that if you treat anti-VEGF, you will prolong survival a little bit. You will definitely shrink the tumor, but you increase the invasive phenotype, and you see vascular co-option. So this is all in laboratory models. And here, if you knock out HIF, which is upstream from VEGF, and so you create anti-angiogenesis in that way, you actually get this dispersion of glioma cells. And then if you add VEGF, you actually encapsulate the tumor, pseudo-encapsulation, but you actually get a sharp border. That was a more recent paper in Cancer Cells. This is something we did at Moffitt and Roskamp Institute. We looked at a novel molecular inhibitor to look at invasion. Of course, there are limitations with animal models, and we want to bring this to the clinic. So here's this is another, so there's a lot of research now on anti-invasion, and I think ultimately we need to have that combination of invasion, anti-invasion, and anti-angiogenesis. And this is a group, Peter Forsyth, I did a very beautiful study in Canada on looking at rational, unbiased search for a molecule that would guide invasion, and it was P75 neurotrophin. And so we started a collaboration with drug discovery folks from Georgetown, looking at this through the Ivy Foundation. So these are today it's well known with the classic paper from Weinberg and Doug Hanahan, the hallmarks of cancer, and angiogenesis certainly there. And we're starting also to look at inflammatory microenvironment and some of the other cytokines, because we're asking the little out-of-the-box question. It's very obvious. You see this every day. My first two slides showed a young person with low grade, and as you get older, what happens? And what happens biologically? And what happens to the cellular environment? So we're starting to look at senescence. And this shows the complexity of the classic paper by Hahn and Weinberg, many of the well-known pathways. But the Judith Campisi's group has shown that as the cells get older, sort of ontogeny recapitulates phylogeny, or so as cells get older, they become senescent, and we usually think of senescence as a very good thing because cells get terminally differentiated and are non-oncogenic. But based on P53, if there are mutations based on the inflammatory microenvironment, you can trigger cancer. So that's a theory that we don't really think about much in neuro-oncology, but something that we're excited about. And it is known, paradoxically, that VEGF, there's more response in those patients over 55. So inflammation is a common denominator in many diseases. It's linked to angiogenesis. There's also stem cells niche, and we know about what happens when you here's a tumor, resected, give Avastin, fantastic result, progression-free survival is increased, and we see this infiltration periventricular pattern throughout the brain, invasion. So, and this was one of our collaborators, Dave Zagzag, and has a beautiful molecular model explaining invasion and with VEGF. And this is John Jane's work on the balance. This also, Judith Folkman had, after we isolated in our lab and in other labs hundreds of, almost everything was a stimulator or inhibitor. We then realized there must be this yin and yang between angiogenesis and its inhibitors and so on. And Tracy Batchelor shown that many of the neuroimaging is the way to get a handle on angiogenesis as endpoints. And we're looking a lot at DTI at our institution at Penn. And this is, well, Howard Fine has shown that this is a beautiful summary if one's glioblastoma and angiogenesis on one slide. Here's tumor angiogenesis, hypoxia, so on and so forth. I won't go through it, but right now, the pharmaceutical industry is interested in glioblastoma, and there's numerous targeted mutations that are possible. And Dave Riedner's calculators like over 1 million combinations. So if you want to go that route, and I think we do need to go that route, there's over 1 million clinical trials. Alan has offered to fund every one of them. So for $100 billion, thank you. And it's possible. Now, but we have to think of some computational, Raleigh's, you have to think of some computational model to sort of predict, because we don't have a million lifetimes to do all this. But now we have actually so many inhibitors. So at that time, the existence of a single molecule that could actually switch on and off was considered, a fantasy in Judith Folkman's mind. Now we have thousands of labs working on this and so many different leads. So how are we going to do with this? And so I just wanted to talk a little bit about senescence. We finally got this thing published because we found IL-6, for example, linked to age, survival, and the grade. And that's one of the inflammatory cytokines, as is IL-A, ICAM. There are other. The paper was published in Journal Geriatric Oncology. One of the authors is one geriatric oncologist. And we sent it to neurosurgery. We thought it would be a slam dunk, right? It's good science. Neurosurgery. They said everything is known about the genomic basis for glioblastoma. There's a landscape, the papers in Science and so on. And, we have to refer to proneural and so on and so forth. Everything that needs to be discovered has been discovered. So there we are. And so you can look at this paper. It's if you PubMed me and so on. So, well, one of the things, going back to Folkman, and our early work is it totally reformed the field of macular degeneration. Used to be a major cause of blindness in the world, totally changed. And Folkman predicted at the time it would. And people, he found only going to cure cancer, he's going to cure blindness. So. But he actually made monumental contributions in that way. And this was from our work where we're looking at the vitreous and we're looking to block angiogenesis. We found an inhibitor, one of the first inhibitors in the vitreous of the eye. And this was a paper from Hopkins. And it confirmed Folkman's concept of tumor dormancy, because as long as the tumor was in the back of the eye without blood vessels, it remained very, very tiny. And the minute it landed on the retina where there was blood vessels, you could see that switch. So I became a believer, and this was when I was a resident. And Henry, who had discovered the first inhibitor of angiogenesis in cartilage, and later took the Hunterian lab, and took it to new heights, was working with me at the time. And what I observed was that it would just float like forever, be dormant. And so this isn't just, go back to those first two cases. This is the young girl who has a low grade, and this is the colonel who has the high grade. It's the angiogenic switch. I'm also going back here, we would see this retinal stalk. We would look at the retina and we'd see a stalk, and in the back of the eye there's this convection flow, and there's a reason why glioblastomas work, goes predictable pathways, and it may have something to do with convection flow. And so this is a paper about how gliadel diffuses out, but this also may be no one's really thinking about why gliomas travel the pathways they do, but they happen to be the white matter, same white matter pathways that subserve function, and there is a circulation there. So I think that's an important area of study. So if you want to read one paper on angiogenesis, it's this paper that Dr. Folkman wrote before his untimely death. And it explains, it predicts why there will be escape, why there's different clones. It talks about promoters and inhibitors. Beautiful paper, Nature Drug Discovery by Folkman, 2007. So sort of back to the future and getting a dose of inspiration. And nice to be back in Florida, by the way, because thank you. And I came here looking for the Fountain of Youth, as did Ponce de Leon a few centuries before. And this is, actually, this week there's a book on innovation, if you want inspiration. Walter Isaacson, who wrote the biography of great people like Steve Jobs and Benjamin Franklin, talks a lot about innovation. And so there's a nice quote by Steve Jobs. And everything at the University of Pennsylvania, every presentation must start or finish with Benjamin Franklin. That's sort of a contract. And so he said, life's tragedy is that we get old too soon and wise too late. So that's a very sentimental talk for me. Thank you, Erica. These are, since I'm in Florida, Al Roten has written this about the brain being the crown jewel of creation. And these are some of the guiding philosophies I have. In cancer research, Peter Huber wrote, the ideal vision of modern medicine includes the surgery with the human body remaining completely intact. What we try to do for our patients is try to, whether it's molecular scalpels or actual scalpels, we try to keep them intact, and I think that'll be the future when we can maintain dormancy. So thank you again. This is what we all do. We look, the brain is a black box. We're going out of the box. Come to Philadelphia in the winter. City of brotherly love. Thank you.